K.DURGADEVI from Department of Biotechnology. Today we will see about PCR and its application. What is PCR? PCR is polymerase chain reaction. It is a medical laboratory technique. It is used in biotechnology and molecular biology lab. It is an amplification process. It makes two single fragment of DNA from multiple copies. So it is called polymerase chain reaction. History. This technique was developed in 1983 in Mullis. He was an American biochemist. Principles of PCR. In this technique based on enzymatic replication DNA, it made to possible generate multiple copies from single DNA fragment, mainly used in polymerase enzyme. It is isolated from Thermus aquatic bacterium. It is a thermostable enzyme, so it makes complete DNA. Next. Components of PCR. These are following components. DNA template. It is an gene of interest. DNA polymerase. It is an tag DNA polymerase. It is used to thermostable, so it not denature at very high temperature. Next one is primer. This is a short single stranded DNA. Next one is deoxyribonucleotide triphosphate. It provides energy for polymerization. Then it building blocks for synthesis of DNA. Buffer system. It used to maintain the optimum condition for DNA denaturation and renaturation. Types of PCR. These are following types: real-time PCR, nested PCR, RT-PCR, multiplex PCR, quantitative PCR. Here RT-PCR used in identified COVID virus. Next one, the following steps are involved in PCR reaction. Here three major cyclic reactions. One is denaturation, annealing, elongation and extension. Denaturation. In this process, the reaction mixture heated 94 degrees Celsius for 2 minutes. In this process, the hydrogen bond break down and separate into single stranded DNA. Next one, annealing process. In this process, the temperature will be slowed down 50 to 60 degrees Celsius. Here this process, we adding primer, it bind to complementary strand, then we can start the DNA replication process. Next one is elongation process. This is a final step of PCR reaction. Here the temperature will be raised in 70 to 80 degrees for 1 minute. Here we are adding tag polymerase enzyme. In this process, finally we get two separate daughter strands. Next one, polymerase chain reaction overall process. This is a denaturation process. We will get rise the temperature 90 degrees Celsius. Here the strands will be separated and we are adding primer sequence. The replication process will be starting. Then we are adding tag polymerase enzyme. Here we are get full daughter strands, two separate daughter strands. Next one, application of PCR reaction. Here the following application used in polymerase chain reaction. One is medicine, testing the genetic disease of mutation and monitoring the gene in gene therapy, detecting disease causing agents in parents. Next application in forensic science. Here used as a tool in genetic fingerprinting and identifying criminal from millions of peoples and paternity test. Research on genetics, compare the genome of two organisms in genomic studies and analysis of gene expression, gene mapping and phylogenetic analysis of DNA. So today we are going to learn about PCR applications.